It's my distinct pleasure to be here. The Montreal Children's Foundation has played a huge role in my career, and I've actually been waiting for this invitation so that I could thank you, but also tell you a little bit about some of the exciting stuff going on in genetics of blindness. I chose two examples coming out of our group to illustrate to you that we are definitely entering into a new era, like Dr. Rosen and Jaquetta Trasler told you. The title of my talk that I chose for tonight is that genetic discoveries in the visual system lead to treatments for childhood blindness, and that personalized medicine is here. Since I started in 1995 at the Children's, I've been studying a disease called Leber congenital amaurosis. This is a devastating disease because it's a congenital form of blindness in neonates. And it's at least 500,000 patients exist in the world. And it affects the retina. Now, what is the retina? The retina is a delicate, multi-layered neuronal tissue that lines the back of our eye, and it captures light that leads to vision, which is then transmitted to the brain and the visual cortex. That is the picture of a retina, a delicate membrane with an optic nerve and blood vessels, and here's the fovea. And when we started in 1995, the reason that I wanted to study this is just to understand it better. But we had no hopes for treatment because everyone told us that this was an untreatable disease. So in 1995, there were no genes for this condition, there were no disease pathways, and no treatments. But thanks to the genomic and genetic revolution, but also because of funding from the Montreal Children's Foundation, CIHR, and the Foundation Fighting Blindness and others, we today have 18 genes for LCA, 7 disease pathways, and 2 treatments. The retina looks like this in a neonate, but also in a young child. And it's in fact a beautiful structure, but it's actually much more complicated. It's a 10-layered structure where the light comes through the retina and hits this very important layer here called the rods and cones and the outer nuclear layer. This is where vision starts. And then the visual information is transferred this way to the ganglion cells, who then send it to the brain. Now, unfortunately, children with LCA look like this. They have tremendous cell death. The rods and the cones are dead or dysfunctional, and they have these huge areas of cell loss. Today, we are at this point in the genetic discovery. We have 70% of the genes known. And so our work is still to find the genes of the remaining 30%. And that's what my first project is that I want to summarize for you, is we want to find the new genes for LCA. We've already found some, but I wanted to give you an example how you find a gene. So we performed next generation sequencing on 60 children from around the world who have LCA, they're blind, and they've been excluded from the 17 genes that we already know. And in 3 of these children, we found mutations in a gene that meant nothing to me a few months ago, NMAT1. So NMAT1 turns out to be an essential enzyme for all cells that is absolutely indispensable to all living organisms because it makes the coenzyme NAD, which is involved in many, many cellular processes, but it also was proven to play a role in neuroprotection. And no human disease has ever been linked to NMAT1. So because of its crucial importance in life itself, we initially did not believe that these mutations could cause only blindness and leave the rest of the body intact. And we dismissed the gene for a few days. We did not consider that NMAT1 was a gene for blindness or for any human disease. Well, we were wrong, because repeat experiments showed the same results. We kept getting NMAT1 mutations in several patients. And then we figured out with some experiments that these particular mutations can kill photoreceptors, but can leave other tissues intact. Thus, we found the first link between this massively important gene, NMAT1, and a human disease, and this human disease is LCA and childhood blindness. Then we discovered that NMAT1 very likely with slightly different mutations, plays a role in strokes, Parkinson's, Alzheimer's, and glaucoma, which is also an eye disease. And the most exciting thing about NMAT1, we believe, is that the treatment avenue are obvious. So sometimes we discover a gene and it's absolutely important and fascinating, but we don't know the function, we have no idea how to treat it. But with NMAT1, already experiments had been done in mice and cells. By upregulating NMAT1, you can rescue a neuron. And you can increase the NAD levels. And this has already been shown. Much more work needs to be done, but it's very obvious how this would lead to treatments. So in my second example, I want to tell you how a gene discovery directly leads to treatment. And this is an example of personalized medicine. Now the gene here is RPE65. It's a very important gene in the vitamin A cycle. Without vitamin A, we cannot see. And what RP65 does, it's one of the control genes in this cycle to make sure that every second we have enough vitamin A to capture light. And what it does is it recycles the photopigment after it catches light. And without RP65 or with mutations in RP65, the child is blind because there is no pigment and it cannot capture the photon. And because of this deficiency, the cell then dies. So we are developing a drug at the Montreal Children's Hospital to bypass the block that these children have. And so we're testing the efficacy and the safety of a small molecule oral prodrug that forms a new pigment in the eye in these children. And we are flying patients in from all over Canada and Europe and China and Brazil with these specific mutations in RP65, but recently also in other molecules of the retinoid cycle. And what we're seeing, to our delight, is that these children are responding to the drug, and we are seeing vision in these children, and we're seeing improvements in the visual field, sometimes spectacular changes, and they have improvements in their daily living, and the drug appears safe. So just to summarize, mutations in this cycle cause it to be blocked so that you cannot make the visual pigment. And without the visual pigment, we cannot capture light. So we're giving a small drug here that mimics this visual pigment. In summary, gene discovery in the visual system leads to identification of disease pathways and insights into disease mechanisms. Two absolutely essential components of understanding why gene mutations cause cell death. But also, when you start to understand the pathway and the mechanism, as I've shown you, it predicts the treatment. And this allows for personalized medicine. And so I was happy to summarize for you two ends of the spectrum, gene discovery and drug discovery, both at the Children's Hospital, thanks to long-term funding, continued MCH Foundation support, international collaborations. And I would just like to say that I'm very proud to work in Canada, because research in Canada and specifically at the Montreal Children's are really great places to do translational bench-to-bedside research. Thank you.